Hi, I'm Naomi, and the assigned cell that I received was the osteoblast. So the osteoblasts are specialized bone-forming cells, and they are found in the bone surface. The main role is for them to secrete a protein called collagen. In my particular cell, I got assigned T1A1, and these form the frameworks for the bone. Minerals such as calcium and phosphate are what deposit into the framework, and this gives the bone the strength. Explaining how the shape of the cell relates to the function, the osteoblasts are cuboidal in shape, and this allows them to line up similar to tiles to like stack on top of each other or alongside each other rather, and it creates the organized layers, and it becomes a base for the nucleus and the organelles. Um, the specific side facing the bone is what secretes the collagen, and it goes into the extracellular matrix. So here we have what I used to make my model. Um, so for the base of the cell, it's just white round cake. For the cytoplasm, which is all around here, it is just vanilla pudding. For the cytoplasm, I use the pudding specifically because it's a gel-like fluid that supports the organelles, and I believe that that was the most appropriate. For the secretion from cytoplasm, it provides just a space and a medium for the ribosomes, for the ER, for the Golgi, and for the vesicles to interact. The nucleus here, I used Oreo, and the nucleus is the command center and stores the DNA, and it regulates the cell activity. For secretion, it holds the collagen genes, and then it is then transcribed into mRNA and starts the collagen production. The ribosomes right over here, which is what I used the colorful sprinkles for, which sometimes do escape from the rough ER, their function is to synthesize the protein. For the secretion process, it processes and it folds the collagen, and then it gets them ready to go into the next step. So from the ribosomes, we go to the rough ER right here. And for the rough ER, I used fruit roll up, and I put a couple sprinkles on there to make it show that it's rough. For the rough ER, no, I'm sorry. From the rough ER to the smooth ER, it is also fruit roll up, and these are the plain ones. And here they store the calcium and they make the lipids, which is important for the bone mineralization. Then from there, we go to the Golgi apparatus, which here is shown as a stack of small wafer cookies. Their function is to package and modify as well as sort the proteins. In the secretion process, it prepares the collagen molecules, and it prepares the collagen molecules into the secretory vesicles right here for the exportation. Secretory vesicles are represented here by the mini M&Ms. Their function is to transport the proteins from the Golgi apparatus into the plasma membrane. And their role in the secretion process is to carry the collagen from outside the osteoblast to form the bone matrix. Then from there we go to the plasma membrane, which is right over here, and it's shown all the way around, and I use Twizzlers as that. For the plasma membrane, their function is to selectively control what enters and what exits. In the secretion process, it fuses with the secretory vesicles, which are again right over here, and it fuses with them, and it releases the collagen into the extracellular matrix. For the mitochondria, which is shown a couple times around here, and for that I used almond slices, their function is to provide energy so that this entire function can keep going. They produce ATP, and their function in the secretion process is again providing ATP for the ribosomes their translation, the vesicle transportation, and then their secretion. The function of the lysosomes is to break down the waste and misfolded proteins. In the secretion process, they are used to destroy the defective collagen proteins, and so they only allow the properly folded collagen to be secreted. Then over here at the end, after all of this process has completed and has done everything correct, over here we have the collagen fibers, and these will lay outside of the cell, and this is what goes to the bone to form. So now just re-explaining everything, I just wanted to re-explain how all that process went through that I just showed you on the model. So the main basic parts of all that whole process is to obtain the nutrients and to obtain nutrients and energy, which is made through the plasma membrane and the mitochondria. The next one would be to synthesize the proteins and other molecules, which would be via the ribosomes and the ER and the Golgi. Then the next one would be to eliminate the wastes. To eliminate the waste, it would be through the lysosomes and the plasma membrane, um, followed by responding to stimuli, which would be the receptors on the plasma membrane, which would be on the outer edge. And then next would be to reproduce. We need the reproduction to continue, adding to that bone and for the bone to complete its full length, which is done through just direct cell division. The nucleus provides the genetic coding. The ribosomes on the rough ER, they translate the code into the collagen protein, and then these proteins are folded and modified in the ER. They're then sent to the Golgi for the packaging. From the Golgi, they go to the secretory vesicles. From the secretory vesicles, they carry the collagen into the plasma membrane. And then from the plasma membrane, that's when it's released outside of the cell, which was what I showed with the cotton candy on the outside of the cell. And then once it's outside of the cell, the collagen fibers just assemble into stronger fibers and they become the framework for the bone. And then through the process, the mitochondria, which I showed 4 different ones around the model, they provide energy, and the lysosomes ensure that it's properly folding the proteins, and they're secreted and they're going to the correct place. So to summarize everything overall that I just explained, osteoblasts are very vital to bone physiology, and without them, the bone couldn't form. It wouldn't be able to grow, and it wouldn't be able to repair itself. Collagen type 1 makes up about, from all the places that I looked up, about 90% of the organic component of the bone. And without osteoblast activity, bones would become brittle and fragile, and things like osteoporosis would occur. And it would just be very painful. Um, all the organelles and everything that I showed just shows how everything cooperates in a single cell and how essential it is to build a strong skeleton, and just making them flexible, functional, and support the entire body. We want to make sure that everything is forming and processing correctly. That way, when we go about our lives, we don't have any pain. Our bones aren't rubbing, our bones aren't breaking, and we just want to make sure that everything overall is just processing the way that it should. Thank you.